Hi everyone, I am the second Marta from Caja Laboratory of Cortical Circuits, and I would like to talk about the multivesicular bodies in the somatosensory cortex. We can see here the reconstruction of 3 dimensions in our samples. Multivesicular bodies, or MVBs, are organelles originated by endocytosis process. They are maturating and they can storage cargo to degrade in lysosomes. They can storage protein, lipids, and it's important because they can storage protein involved in Alzheimer's disease and Parkinson's disease. And then MVBs can fuse with cell membrane to produce exosomes. And they can recycle proteins to the cell membrane again, like receptors, synaptic receptors, and they are involved in some process like long-term potentiation. And they can fuse with other organelles that are involved in autophagy process to degrade the cargo again to the lysosome. And we use a brain of rat that we process for electron microscopy, and then we obtain our images with a fifth microscope. Then we have to analyze these images with SPINA software, and I would like to show an overview of the function of SPINA. Here we can see the image that we obtained with the electron microscope, and here is an MVB that we can segment it with tools that provide us SPINA. Here in pink is in 3 dimensions segmented, and we can visualize with the 3D view. Here we can see the synapse and the MVB in pink in 3D view. And then we can obtain the size and the spatial distribution. And we found MVBs in dendrites in the postsynaptic fiber. As we can see here, this is the synapse, and here we have the MVB. And we found MVBs in the presynaptic fiber in axons, very close to the synapse. And we found that it's more frequently found the MVBs in dendrite fibers than in axons. And these MVBs are bigger in the dendritic fiber than in axon fiber. And it has been described in the literature that MVBs are related to mitochondria, but we quantify that more or less the 20% of MVBs present this relation. We can see here the MVB dock to mitochondria, and they are storing the cargos that are oxidated by the metabolomic process in mitochondria, and they can maintain the function of mitochondria. And these MVBs are larger too than the undocked MVBs, because they are storing all this cargo. We observe other kind of MVB, called a tubular MVB, because present tubular extensions. And we can see here the image that we obtained, serial image of this MVB, and here the 3-dimensional reconstruction. And this MVB is the 7% of the total MVBs that we studied, and are larger to non-tubular MVBs. And it's related, this state is related to the recycling process that I described before. And then we observe the spatial distribution of MVBs in our samples with some statistical tools like G function, F function, and key function. And we observe that most of MVBs are in a random distribution in the spatial, but the 30% of these MBVs are in a cluster distribution. In summary, the MBVs located in dendrites outnumber axonic MBVs in a proportion of 2:1. And 90% of MBVs are in direct contact with mitochondria. The tubular MBVs are 7% of the total MBVs that we studied. And the MBVs are now randomly distributed in 70% of our sample, but they are in a cluster distribution in the remaining 30%. And thank you very much for your attention.